Okay, yes, so the topic I want to talk about today, interactions of cyanobacterial blooms with energy fluxes at the air-water interface, is kind of a relatively new topic to me, and I thought I can use that seminar that I give at EPFL to also start some cooperation with you guys over there, because I think there are a lot of things to be explored in that regard. I want to start my presentation with referring to a very old paper published by Johnny Wüsten almost or more than 25 years ago. That paper was only published in German language. The translated title is Interactions in Lakes, Biology as a Source of Dominant Physical Forces. And in that paper, Johnny analyzed processes or mechanisms in which physical or the biology in aquatic ecosystems change or affect the physical setting of that system. By the time that paper was published, I was graduating actually, so I just started to become interested in physical processes in lakes and reservoirs. And for me, that paper was very inspiring, and actually, I think it influenced me a lot. And I was very happy that I had also the chance to work on some of these topics that Johnny was analyzing in that paper during my time at ERWAC when I worked with Johnny. And one of the topics he introduced in that paper was the so-called bioconvection, that is the physical mixing of a density stratified water column by tiny swimming bacteria. And as I said, that was more than 25 years ago. And apparently it was not only me who became very fascinated in these topics, because also Johnny published actually a whole series of papers in the recent years on that topic and explored that much further into a very to a higher level of detail. However, this is not what I want to talk about today. Today, I want to talk about cyanobacterial blooms. And cyanobacterial blooms, or harmful cyanobacterial blooms, harmful because they produce toxins. It is a very widespread and increasing phenomena that can be found in lakes and reservoirs and ponds and many aquatic systems worldwide. You probably all are aware of these blooms, and one of the most annoying features of these blooms are the so-called surface blooms or scum layers, which is a huge accumulation of these usually planktonic algae at the water surface, where they produce this scum layer or a foam being on the water surface. It is not a region, it is a global phenomenon, and generally these blooms of toxic cyanobacteria, they are a growing threat to freshwater ecosystems because they can completely change the ecosystem where they appear. And it is also a threat to water resources because they strongly interfere, for instance, with drinking water production in lakes or reservoirs. There's still a lack in mechanistic understanding of how these blooms actually form, and specifically how they can persist at the water surface over longer times. And of course, there has been done a lot of research on the environmental factors that control cyanobacterial blooms, and one of the most important properties is that they can actually fix inorganic nitrogen, so they do not rely on nitrate or ammonia, and so they are especially successful in aquatic systems where we have high phosphate levels and rather low nitrogen levels. Under these conditions, they can outcompete other algae. Now, the physical properties of the aquatic system that affect the occurrence of these cyanobacterial blooms is turbulence. They only occur, or they mainly occur at low turbulence conditions and under high light conditions. And conversely, if there's high turbulence and strong vertical mixing, this negatively affects these blooms, and usually these blooms disappear after a wind event or also after a rain event, which causes quite some mixing in the water column. Now, consequently, the strategies for preventing these blooms, they have nutrient management at the first place, so reducing nutrient inputs, and at most places where these blooms occur, their occurrence or their frequency of occurrence has been linked to eutrophication. And other strategies include chemical control, like the precipitation of phosphate or other chemical controls, removing either the nutrients or the algae from the water column, manipulation of the aquatic food webs, or artificial mixing of the lake or the reservoir to get rid of these blooms. Now, these strategies for prevention, they all work more on the long term, and for a short-term control where people just want to get rid of these blooms, there are also a huge variety of techniques applied, including ultrasonic treatment of the lake water or also UV treatment of the entire lake water in order to kill the algae. And where I became aware of the huge economic value or the huge economic damage that these blooms do. This was in China. That picture you can see here is from Lake Taihu in China. This is one of the largest shallow lakes, and it is used as a drinking water supply for the Shanghai metropolitan area. This lake is severely affected by cyanobacterial blooms, and what they do there, they pump these blooms out of the water. So they have these pumping stations, they have vessels crossing the lake, filtering the lake water, removing tons of algae just to remove the algae from the lake water before the lake water gets treated for drinking water production. So as I said, artificial mixing is a strategy to prevent or get rid of these algae blooms. What does mixing do? So the mixing is often done in terms of bubble plumes. So air bubbles are injected at the bottom of the aquatic systems. They rise to the surface, and there they mix the water column completely. And at the end, these artificial mixing, it can increase the nutrient concentration by resuspension and mixing with the lower layers of the water body. It decreases the stability of the water column. It decreases the light dose that phytoplankton receive just because they are continuously moved through that strong vertical light gradient. At the end, the outcome of that mixing is that the phytoplankton biomass can actually be increased due to the mixing, due to the higher nutrient concentration. However, the competition between cyanobacteria and other algae, including diatoms or green algae, is more on the side of the other algae. So that's a way to get rid of the cyanobacteria. However, this artificial mixing can be applied in small or medium-sized lakes. It is quite expensive to operate, and of course, that would not a feasible method to remove these algae from larger lakes, including the Lake Taihu that we saw on the other slide before. Now, another important properties of many of these cyanobacteria is that they can become buoyant. That means they have a density that is below the density of water. With that, they rise to the water surface, and there they form these surface blooms or these scum layers, these dense layers right at the water surface. And these are the layers that are of most concern, because in these layers, the toxin concentration can be very high. They can become harmful for larger animals or even for human consumption in these dense scum layers. Where the buoyancy regulation in these cyanobacteria is quite well studied. However, as I will show you in a minute, the formation and the persistence of these scum layers in relation to the physical forcing is still rather poorly understood. So what happens, what makes them buoyant, it is positively or negatively buoyant. It depends on light exposure. So during the exposure to light, the rate or the density of the cyanobacteria is increasing. It increases because they accumulate carbohydrates in the cells. And oh, that's actually wrong. So it is not a gain in buoyancy, it is a loss in buoyancy. So during light exposure, their density is growing and they start sinking from the surface to larger depths. Now, in darkness, either at night or when they have arrived at larger depths, at a larger distance from the surface, their density is actually decreasing. So the rate of density change becomes negative. That is caused by the consumption actually of the carbohydrates that they have incorporated under light conditions. And then they gain buoyancy. So these two, I will say, are mixed up and they rise to the surface again. And this buoyancy control just, leads to the so-called diurnal migration of microcystis, one of the most common cyanobacteria species that is doing this migration, that is regulating buoyancy. And what you can see here is a simulation which combines actually this buoyancy regulation with a physical forcing. So we have the solar forcing, the solar radiation here with a dial pattern in light and darkness at night. Here in the middle panel, we see the actually path of a single cell. So as soon as the light starts to increase, the cells sink because they accumulate the carbohydrates. However, if they reach larger depths, then the light intensity is reduced due to attenuation in the water column, and they start rising again, arriving at the water surface typically at night. And on the next day, when the light starts again, they are removed from the water surface because their buoyancy is increasing again. And with these buoyancy regulation and the physical forcing that causes the mixing of the water column, it is the diurnal migration could be described quite well, like shown in that paper of Evelyn Aparicio-Medrano, where also Rob Uettenburger was involved. And we can basically follow this migration of the microcystis or other cyanobacteria. This is kind of a reversible migration because they would move up and down through the water column in response to light. However, these scum layers that I showed you before, they are also persistent during daytime in some systems. And this cannot be explained by that model because according to that model, during daytime, cells that are sitting at the water surface would do photosynthesis, they would gain weight and sink down from the water surface. They would typically disappear during daytime. So these scum layers, they cannot really be explained by that model. And the question is, are there any additional interactions between surface blooms and physical processes near the water surface? And this is what I want to talk about today. And first, I want to introduce a well-known mechanism that is self-shading, and then coming to another mechanism that is that is the presence of the air-water interface. Surprisingly, self-shading, that is the change of the attenuation, of the light attenuation in the water column due to the presence of the algae, has not been considered in this model so far. And it has mainly not been considered because the simulations were done with much lower cell densities than we can find in these scum layers. And so our hypothesis was that there is probably a feedback regulation. So if the microcystis cell density is increasing in the water column, that causes higher light attenuation. And if there's less light, then the microcystis, the cyanobacteria, will gain buoyancy and rise to the water surface. So as more microcystis there are, as more the cells can be expected to be within a narrow layer near the water surface because of the strong light attenuation by the algae cells. And so what we did, we measured the light attenuation by microsister cells and lab experiments. So we estimated the concentration-dependent light attenuation coefficient, and then we simulated light cycles with the presence of microsister cells. And depending on the concentration of these cells, you can see when there are no cells in the water column, we have penetration of light to large depths. If we increase the cell density to up to 10 to the power of 10 or 11 cells per liter, then there's basically no light below a very thin surface layer. We then added this buoyancy regulation model that has been used in the other studies before and incorporated the self-shading by the algae. And the effects you can see here in this matrix, this is showing light intensity first. And in these graphs, the colonized size is increasing from left to right. And the cell density is increasing from top to bottom. And these are the results of these numerical simulations where we disregarded any water motion for the moment. So it's only buoyancy regulation and light intensity that we consider here. And what you can see, if we have large colonized and high cell densities, the cells will stay at the water surface all day long. So there's no diurnal migration anymore. Why is colonized size actually important? The colonized size, so these cells aggregate to colonize, and as larger the colony, as higher is the sinking or flotation velocity. This is Stokes' law, so very small particles sink or rise very slowly, while larger particles sink or rise in the water column more quickly. Now, if we now look at the distribution of the microsister cells, and for low cell concentrations, in these simulations, we started with uniformly distributed algae cells over a layer of a few meters of water. We can see all these figures show a diel light cycle. So the light starts at 0 hour, is maximum at 6, and there's darkness after 12 hours again. At low cell densities during the light cycle, the cells sink farther down. This is what has been predicted by other models. However, if we increase the cell density, and the different colors show again different colonized sizes. So for large colonized and high cell densities, the cells rise up to the water surface and they stay at the water surface. The reason is that there's very low light intensity due to the strong self-shading by the cells. Now the effect becomes much more pronounced if the cells start their migration at the water surface. So if they are already at the water surface by the time the light starts to increase, then we can see that already for lower concentration and for smaller cells, the cells or the light penetration is more or less inhibited by the presence of the cells. There are additional interactions among different colonized sizes because, as I mentioned, they have different rise and flotation velocities. So small colonized sizes, they will sink more slowly when the light is switched on, and they provide extended shading for larger colonized that sink more rapidly when the light is switched on. And in the darkness, the larger colonized, the larger cells rise more quickly to the water surface again. Now, what these simulations showed, we had no measurements except for the extinction coefficient, is that self-shading that has been completely neglected in these simulations so far is a potential mechanism that can explain the persistence of cyanobacterial blooms, of surface blooms during daytime. What we did not consider are water motion, of course. The question is, to what extent can that surface scum layer resist wind-driven mixing during daytime? Now that is then kind of the topic of the second part of my talk, and that study somehow started when I became interested in these microcysters. I had that small aquarium on my desk with a microscopic camera just to learn a little bit more of these cyanobacteria, and whenever I had time during the day. I was watching how they are doing and what they do. And what I noticed is when that aquarium is standing on my desk, then there's always some motion, of course. These are convective currents due to temperature changes in the room. And the cells, they are always in motion below the water surface. However, here in the upper part of that video, you can see the actual water surface, and that water surface is not moving. It's completely stagnant. And by looking at that in more detail, it really appears that the water surface has more the impression of a solid boundary of the water body. Now, if we have a detailed look at the water surface now we have a camera looking at the water surface, and what we can see, these focused cells that we can see here, they are sitting right at the water surface, and the more blurry cells that are moving very rapidly below the surface, they are at larger depths. That's a time-lapse video, so these slow convective motions, they now look very rapidly. And what we can see is that these convective motions, they actually transport algae cells to the water surface. At the water surface, they got trapped, and they accumulate at the water surface and form these large aggregates that we can see. Okay, that I found very interesting. So we went to the lab with that setup and had a more sophisticated look at that. We developed a wind-driven mesocosm system to study that process, to add also wind velocity and wind-induced mixing. And we used an annular flume. Basically, it's a circular wind tunnel, which was filled with 30 centimeters of water or 40 centimeters of water, and we could drive a flow using small fans that created a wind blowing across the water surface. Then we were following the accumulation of cells at the water surface. So we had a microscopic camera looking at the water surface, looking at the cells, looking at their velocity, and looking at their size distribution over time. And we had a second observation in the bulk water. There we used a laser light sheet to illuminate a narrow range of the water column, and we followed the cells. Again, we measured their velocity and we measured their size in the bulk water below the surface. Then we simulated diurnal cycles and wind speed in combination with diurnal cycles and solar radiation. So as a typical feature in many lakes, we can observe that wind is increasing over daytime and decreasing or is very low during night. We used that pattern and followed the bloom formation under these conditions. And during a 3-day experiment, we can see that these are images looking at the water surface from above. So we look at the surface bloom. We can see that the bloom is forming over the period of 3 days. The algae become more and more dense at the water surface. And the surprising finding here was that wind actually did not have any effect apparently, or almost no visible effect. We increased the wind up to more than 3 meters per second because 3 meters per second were reported as a threshold wind speed for the occurrence of surface blooms in lakes. And while during the first day we could still see, okay, when we switched on the wind, then some algae disappeared from the surface. But actually during day 2 or day 3, when we had these dense algae, they were not very impressed by wind at all. So let's have a more detailed look. This is the surface coverage, and the different colors in these box plots. They show you different wind speeds. Again, on day 1, day 2, day 3, we see the general increase in surface coverage over time. And during the first day in that bar, we can see that the surface coverage by algae actually slightly increased with increasing wind speed over day. On the second day, we started with 0 wind here. Then we had the highest wind during noon that slightly reduced the surface coverage, but it quickly recovered then in the afternoon when the wind was going down again. On the third day, with this dense algae, again, we saw a slight decrease in surface coverage, but the surface bloom very rapidly recovered. In the bulk water, below the water surface, we saw kind of the opposite. So during the first day, the concentration in the bulk water strongly decreased. these are cells that were accumulating at the water surface, promoted by the wind, by the motion. On the second and the third day, we saw, okay, when the wind was high, like here during midday, we saw higher concentration of colonized in the bulk water. These were the few colonized or cells that had been mixed down by the wind, but not very dramatic, that increase. If you look at the colonized size distributions at the water surface, then on day 1, we had rather small colonized, but up to 2 millimeter in size. And then on the day 2 and day 3, we observed these large aggregations being up to 6 or more millimeters in size. And when we increase the wind, so the different colors are different wind speed, we can see these large aggregates, they persist up to wind speeds of 2.5 meter per second. For 3.6 meter per second, these very large aggregations disappeared. They were broken down to smaller aggregates being, yeah, more or less smaller than 2 millimeter in size. Now, the most fascinating result, however, was that there was a suppression of momentum transfer from wind to water by the presence of the scum layer. What you can see here is the flow speeds that we observed in the water near the water surface as a function of wind speeds that we applied above the water surface on the 3 days. And on that first day, day 1, we had this quite low coverage, very low algae concentration. And on that day, for a wind speed of 3 meters per second, for instance, we had a flow velocity near the water surface of, yes, something around 7 millimeters per second. On the second day, with a more dense coverage of the surface by algae, the same wind speed caused a flow velocity in the water, which is only 50% of that. On the third day, with a strong coverage, it was further reduced to about 1/3 of the flow velocity that we observed at the same wind speed without algae. So obviously, there's a stabilizing feedback mechanism of the surface bloom with its physical environment, because we saw at the beginning, these blooms, they love low turbulence conditions. Wind is generating turbulence. The aggregation of the cells near the surface, they reduce the momentum transfer from wind to water, so the same wind speed would create less turbulence when the algae are there. And to give you an idea, so these are video recordings taken looking at the water surface from above. Okay, this does not really work. So you can see, so you can't see it, but what you were supposed to see is that on day 2, the algae or the surface layer was still moving at the wind speed of 2.5 meters per second, while on day 3 at 2.5 meters per second wind speed, the surface was basically not moving at all. So what we found was that the surface scum was promoted by the aggregation of colonized from the water column at the water surface. So these large aggregations, they were only formed at the water surface. And these aggregations, they determined also the stability of the scum layer against wind forcing. The presence of the scum increased the threshold wind speed for the onset of flow. So for instance, on day 3, there was almost no motion of the water up to a wind speed of 2.5 meters per second. And even at high wind speed, it strongly reduced the turbulence and the flow velocity in the water. So there's a stabilizing feedback mechanism of the algae with its physical environment. Now, what are the implications for that? And also more importantly, what is the mechanism behind that? Generally, the magnitude and the dynamics of wind is a very important physical feature for the functioning of aquatic ecosystems, especially lakes. It plays a vital role in ecology and biogeochemistry. Now, the surface or the turbulence produced by wind in the water column, that can be described by the surface, the shear stress at the water surface, and from simple bulk scaling, the shear stress at the water surface is related to the wind speed, this is U10, by a so-called drag coefficient. And the shear stress, the force created by the wind blowing over the water on the water surface, scales with the square of the wind speed. Now, what is the mechanism that causes the reduction of the surface drag coefficient in our experiments? The surface drag coefficient, that brings me to another open research question that Johnny worked on many years ago, that is the wind speed dependence of the drag coefficient at low wind speed. So that drag coefficient has been found to depend on wind speed. It has a minimum at wind speeds around 3 to 5 meters per second. It slowly increases for high wind speed. This is because the drag coefficient is mainly determined by surface roughness, the roughness of the water surface. For high wind speed, here we have long gravity waves, high gravity waves that increase the roughness. At very low wind speed, we do have capillary waves, these small ripples that you can see at low wind speed on the water surface with some centimeters or decimeters in wavelengths. These are capillary waves, and they increase the roughness of the water surface disproportionately strongly. That was one of the assumptions, or one of the hypotheses of what is causing that increase in drag coefficient at low wind speed. However, what is the mechanism that is related to that presence of the microcystis? Because the algae, they were not on the water surface, they were still below the water surface. So they did not change the roughness of the water surface due to their presence. How could they change the drag coefficient? And one of the hypotheses that we had is that they actually changed the surface tension of the water. And with that change in surface tension, there's a damping of capillary waves. That means at low wind speed, no ripples will be formed anymore, because the restoring force for these capillary waves is surface tension. If surface tension is reduced, there are no capillary waves at low wind speed, and this potentially reduces the drag coefficient. Now, unfortunately, we were not expecting these results in our experiments, and therefore we did not measure surface tension in our original experiment. However, after that experiment, we measured surface tension in microcystis suspensions, and we found that indeed the presence of microcystis decreases the surface tension of the water significantly. So that links the mechanism of drag reduction by microcystis to the so-called surface microlayer. The surface microlayer, that is a research topic that has been receiving strong scientific interest, mainly in the marine environment in the recent years. The surface microlayer kind of is actually a whole hierarchy of layers that starts with a very water surface where we have surfactant, surface active material that change the surface tension of the water. Below that layer, we have accumulation of all types of solutes, but also of bacteria and other organisms. As I said, the surface microlayer has been receiving a lot of attention in the marine environment. This is where people actually also related the existence of surfactants of the surface microlayer to a change in water surface roughness that has been observed from radar satellite observations, where people found wherever they have large phytoplankton blooms in the ocean, they see less water surface roughness at the same wind speed. And this has been related then to air-water gas exchange, the surface microlayer, so that film reduces gas exchange, but also the reduction in turbulence due to the reduction in drag coefficient causes a reduction in gas exchange velocities. The uptake rate of CO2 in the ocean, of course, is of great interest because the ocean is a great sink for CO2, and the rate at which CO2 is taken up by the ocean is strongly controlled by the gas exchange velocity. And there have been also evidence from the literature that it's actually the EPS, the extracellular polymeric substances, that is produced by microcystis during bloom formation, So far, it has been believed that these EPS are produced in order to promote colony formation. So the cells produce these EPS, these sticky mucilage, which then makes the individual cells sticking together and forming larger colonies. However, there have been no direct link being made between these surface microlayer and cyanobacterial blooms, especially not in inland waters. That's kind of an ongoing, a current research topic where we explore this at greater detail. So kind of repeating the experiments that we did with these wind-driven mesocosms, but at the same time also take care of what is the biochemical composition of the surface microlayer, what is the surfactants, and what is the drag coefficient and the roughness that we see at the water surface. Yeah, very quickly, I want to conclude. So there are still many challenges in understanding biological physical actions in aquatic ecosystems. There are still popping up new types of interactions. And what I find a very interesting or very important research direction for the future is actually the interactions of surface films, of surfactants, of the surface microlayer with hydrodynamic processes in inland waters. And with that, I want to thank you, and I want to thank those people who were involved in the work that I presented, and the DFG for funding it. Thank you.